Okie dokie, so today we're talking about leading strand replication. And so I'm going to talk about kind of the proteins that are involved in that and everything. So the first protein, and so right here I have my replication bubble. Right here it's the replication fork, my strand of DNA. So here it's kind of untwisted. Right here I'm going to have my DNA. So the first protein to know would be DNA helicase. And so this is the unzip, cut open, the scissors, whatever you want to call them. I think of a helicopter. Helicase helicopter. And so a helicopter has blades, and that would catch you. I think the reason it's called helicase is because this is a helix, and case for enzymes breaks apart, and so we're breaking apart the DNA. But we don't want to really break the whole thing apart, and so that's why we're going to have our single-stranded binding proteins. Single-stranded binding proteins. And so these I think of like in surgery when you pull something apart. Basically, when you pull something apart and you gotta hold it apart because it wants to stretch back together, that's what this does. Because the DNA is in this twisted shape and it's really tight, and then with the DNA helicase, so I'm gonna put an HELI here. It's trying to do so. Single-stranded binding proteins, SSBP. The helicase is cutting it apart, and so we want to keep it apart, and so we're going to pull it apart with the single-stranded binding protein. So keep apart. Okay, so then, cool. So that's kind of the machinery that's taking the DNA and it's coiled up and uncoiling it, unzipping it open. Cool. So now this is the leading strand. So what's going to copy the leading strand is going to be DNA polymerase 3. So DNA polymerase 3, it's going to read from 3' to 5'. So I always think reads toward the biggest because this gets complicated really quick as far as when you do the replication bubble that we looked at. And you have all these 3' and 5' running around, it's kind of get jumbled in your head. So I just think it reads towards the big number. So read towards the big number. Read toward big. And so here I have my 3' end. Down here, if we follow the thing, my 5' end, so 3'. I know it's going to be 5' right here. And so my DNA polymerase is going to be, I'm going to put them right here. So DNA polymerase, or poly 3, is right there. And so he's going to be putting on the bases here. And so as he's putting on the bases, he's building a 5' to 3' strand. So they build in the anti-parallel. And so that's what it's called when they're like this, and one runs this way, the other runs that way, or vice versa, anti-parallel. Because they're going like this. Not together, but apart, or like that. Either way, so 5', 3', and so I'd have my 5' end here. DNA polymerase is just going to move on that way, and that's really all the machinery it takes for the leading strand. Is just the stuff to open it up, and then the DNA polymerase 3, and it's just going to keep chugging along and keeping up with the replication fork, and that's about it. Thanks for watching.